Hello everyone, welcome to InSciLabTech community. I'm Sonam Kanchan, a PhD graduate. As part of our For Researcher by Researcher initiative, I'm here to show how to calculate and prepare graph for percentage cell viability by using Excel. Before starting, we need to know what is cell viability and why we require percentage cell viability. Cell viability is a measure of the proportion of live and healthy cell within a population, and we need to check percentage cell viability when we require to check the effectiveness of any drug against the cells, which may be cancerous cells, mammalian cells, or bacterial cells. For this, we need to perform the cytotoxicity assay. Today, I'm going to tell you how to calculate and prepare graph for percentage cell viability. Here the percentage cell viability is equals to sample OD minus negative control OD divided by control OD multiplied by the 100. This is the formula for the percentage cell viability. We have various drug concentrations such as 0, 20, 40, 60, 80 and 100, and the replicates R1, R2, R3. These are the three replicates from our experimental data. But what we next require to get the average value, simply put the equals to mark and find the average function and then select these values and then enter. Next, we simply drag it down to get all the average values. Then what we next need for percentage cell viability, we need the sample OD minus negative control OD. This is our normalized or final data. Sample OD is the optical density. For our sample, we simply put equals to mark, select this and subtract it with the negative control value to normalize it. We do the same thing for each and every value to get the normalized or final value. After getting the final OD, we get the cell viability by dividing it with the control OD. Simply equals to mark, select it, divided with the positive control OD. Do the same thing for each and every cell to get the cell viability or K value. After getting the K value for percentage cell viability, we simply multiplied the K value with 100 equals to, drag it down to get all the percentage cell viability. Here we get the all percentage cell viability and drug concentration. We require drug concentration and percentage cell viability to prepare our graph. Simply go to insert, then recommended chart, select the chart which we require. Here I am using this bar diagram. To edit the chart, we have add chart element option. Simply select, and from here we can add axis title, chart title, data labels, error bars, grid lines, legend, trend lines. To editing the chart, we first need to select it, and then what we need to mention, we just simply mention it. We can add error bars here. For error bars, we require SD, standard deviation or standard error. To check how to add error bars, please follow and check our next video. I hope this tutorial was helpful for your research. We would also love to hear your feedback directly. Let us know what you think in the community. To continue the conversation, ask questions, or collaborate with the fellow scientists, please join our community hub at InSciLabTech. The link is in the video description or pinned comment. Thank you. Have a nice day.